Okay, so in the video today we will see how MACS is able to identify peaks. Now let's remember a little bit how ChIPSeq works. It comprises actually 4 main steps. In the first step we cross-link the protein with DNA. So if this is a protein here, this one, we make it to be firmly attached to the DNA that it binds to. Then we break the DNA. So if this is the whole chromosome, we make some breaks, for example here. Then we put some antibody to precipitate this piece together with the protein of interest and the antibody. Then we purify the DNA, which means that we just keep this part of this complex, which is the DNA, and then we sequence that. So let's assume that we have a tissue with just 4 cells. Then in the cell number 1 we have precipitate this complex, cell number 2 this complex, cell number 3 this complex, and cell number 4 this complex here. Now if we sequence that, then from this piece of DNA we might sequence this part, this 5 prime, from this part. Perhaps from this one we will sequence the negative strand, so we will sequence the 5 prime end of the minus strand here, and let's say that for this one we'll sequence also the minus strand. So if we sequence many different parts of the DNA that correspond to this protein, then we will get either reads from here, which will correspond to the 5 prime end of the negative strand, or we will get reads from here, which corresponds to the 5 prime end of the positive strand. Now if we will map these reads back to the reference genome, so if this is, let's say, the location that the protein binds to, then aligning these reads we will get pieces here, so reads here, or we will get reads here, and we'll get reads here. Okay. So a real target of the protein is flanked by a lot of reads on one side and a lot of reads on the other side. So there is a whole region here, like this region, that contains many reads actually. Okay, let's now see how to identify these peaks. And the first step, step number 1, is to identify the so-called interesting regions. So these are not yet the regions with the peaks, but among them we might find several regions that correspond actually to peaks. Okay, so let's see how to be able to find these interesting regions. Now if this is a region that corresponds to a peak, we see this characteristic bimodal distribution of reads. So we have many reads here which form a peak, and then later on we have another region that forms another peak. Okay, so if this is the distribution of reads in an interesting region, then we can do the following trick, and actually MACS is doing that, in order to fill, let's say, this gap, and also have a higher depth, higher coverage here, which is what we expect, in fact. So to be able to identify the central region where the protein binds on the DNA. So we know from the experiment that the fragment size that we expect to have, this is due to the truncation, is let's say something between 150 to 500 base pairs. Now let's just assume a value here. Let's assume that this is 210. So from the truncation we know that the fragment size that we should have should be around 210 base pairs. What we do is that we extend every read here towards the 3' end, so this one is extended up to this point, and now this part here is 210 base pairs. And we do the same for all reads on this side. So the 3' end, remember that this is the 5' end of this read, so the 3' end is here. Now again this one will be 210 base pairs. Okay, by extending all the reads towards the 5', then what we will eventually manage to have is more reads, higher depth actually here, because we have extended everything. And from this side. Now why we do that? Because the next step will be to scan the whole genome using a sliding window that has approximately this size, 210 base pairs. They identify interesting regions. The problem will be that if we have these reads, let's say these ones, and then a bit further these ones, then the window that we assumed in the beginning will not be able to cover both of them, so it might be like that. Now let's say that this is a bit further, and then when we move here, it will not catch both of the peaks together. Then when we move here, we get only this one and not this one, and so on. But by extending the reads here in the middle and having from both sides higher depth here, then as long as we are here, we will catch this one, and then here we will catch, because we'll get this middle region here and here and here and so on. Okay, so we have extended the reads, and now we don't have this bimodal distribution any longer, but what we have is, I will just use one color now, regions like that. High depth here and lower depth here, after the extension is that. Then we have some noise here, some reads that do not form a peak, and then perhaps here we have again such a significant region, and so on and so forth. Now we use a window approximately of this size, so we scan it. Let's say that this is the window. We identify the number of reads that are inside here. Let's say these are 30 reads, and then we move on one base pair to the right, so this is the next window. Count the reads, so nothing will be excluded here, but some new will be entered here. So let's say that this has 35 reads, and then we move one position. Now some reads are excluded perhaps, and some other included. Let's say that in the third one we have 38 reads, and therefore we can form a series of reads inside such a window of 210 base pairs. So let's say that this series of numbers is 30, 35, 38, 41, and then 39, and then 37, and then let's say that after that we exit this region, then we will have 8, 7, 6, 5, and so on. Then at some point we start to have again a lot, 21, 28, 35, and so on and so forth. Okay. So if we continue that, we might have a long series of numbers that each one represents the number of reads in such a window. Just a small correction here, because I made a small mistake. If this is the fragment size from the sonication, actually we use the double size of this, so it will be 210 multiplied by 2, which will be 420 here. A small technical detail. And then we do exactly this work with this window size, and we slide it along the genome. We count in each one of them how many reads we find, and this is just an example here. And in the first step, what we identify are regions that have 10 to 30 times more reads than average. So if all the windows on the genome, let's say they have an average of 7.8 reads, this is an average value, then we will consider as interesting reads in the beginning those regions that have 78, 10 times this number, up to 234, if I have not done any mistake here to this multiplication, up to 234 reads. So again, this is the average along all the windows in the genome, and these are the reads which corresponds to 10 times more than the average, up to 30 times more than the average. Okay, and let's say that we have now such a let's say that we have identified, I will just put a number here, 50 such regions. And let's say that one of them is this one. Remember, we have extended reads like that, and we have also the other one from the negative strand here, and we have extended these reads towards this direction. And so what we will do now is the following. We will see separately the reads from the plus strand and the minus strand. Now the reads from the plus strand have this location, so the average, now I'm not very sure from the manual, but maybe it's not so important which part of this read we take. We might take the middle, or we might take the beginning or the end. Let's say that we take the beginning. So we take the beginnings of all reads from the plus strand, which are here, and we find the average location. So you have to average between this number here and this number here and this number here and this number here. So these are the locations. These dots, black dots here, are the locations of the beginnings of each read as they are mapped on the reference genome. If we find the average of these values, it might be this location here. Now we do the same from this side. We take again the beginnings, we find the average. Let's say that this average is somewhere here. Again, I'm not sure from the manual if it takes, but it's not so important, if it takes the beginning of each read or if it takes the middle of each read. Let's say that it takes the beginning of each read. Okay, so the distance between this one, MACS call it D, and corresponds to the fragment size. And it does exactly this work from all the regions that have from so many to so many reads. So we said that let's assume that we have 50 regions that have 10 to 30 times more reads on the average, 50 regions. In each one of them we find the beginning and the ends. So if this is the first one, we compute a D1 for the first region, and then for the second one, the second region, again we find the similar stuff. I will just not use colors now. These are the plus and the minus, and here we have D2, and therefore we have 50 such numbers because we had 50 regions. So D1, D2 up to D50. And then from these D's we find the average value. And this is the so-called fragment size. Okay. Okay, I just read in a paper that instead of taking the beginnings here, it might follow the following procedure. So to find how many reads we have here, how many reads we have in each location, and therefore make a distribution of the number of reads in each location. So for example, here we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 reads. There are more reads here from the plus strand. And somewhere here we might have more reads, so another distribution here. And what it takes is the position of these reads, is not the beginnings, as I said previously, but the position that corresponds to the highest value of this distribution. It's not super important for us. In any case, it follows this procedure that is not so difficult to demonstrate, that it finds the location of one peak and the location of another peak, and the distance between these two locations is called the fragment size. So in any case, we have the fragment size, and let's say that we said that this is the average value of all these fragment sizes from all the interesting regions, and let's say that this D equals to 250 base pairs. So again, to sum up what we have done until now is that we extended each read from the plus strand towards the 3' end until to reach the size of what we have sonicated, and we said that let's say that this is 210 base pairs. So we extended the read computationally. We extended the read from the negative strand, again computationally, and then we use a window size of 420 base pairs, so 2 times the size. It's not very nice that we use the term fragment size here. Let's use the term shearing size. Okay, and then we scan the whole genome. We find regions that have we calculate how many reads we have in every window, and let's say that these are the numbers in every window of 420 base pairs, and then we identify regions that have 10 times to 30 times more reads than the average. Yeah, then MACS, oh, I made one more mistake. I didn't describe the whole procedure in detail. So what MACS is doing then is that does not take all these regions, because it seems all these regions that have 10 to 30 more reads than the average are several thousands. So let's say that these are 5,000 regions. Randomly selects 1,000 of them. So this 1,000 is this 50 that I used here, which all of them have 78 to 234 reads, so 10 times to 30 times more than the average. Now for each one of this region, again, it takes the different modes of the distribution, taking separately the reads that correspond to the plus strand and the reads that correspond to the minus strand. It finds the middle location, or the location that corresponds to the peak of this distribution for the plus strand, and this location that corresponds to the peak of the negative distribution of the depth from these reads, and this distance is called di, where I is the corresponding fragment. So here we have 1,000 fragments, 1,000 such regions. Again, we do the same for the second location, third location, up to 1,000. I should correct also this one, just to be accurate according to the manual. And then it finds the average value of the D here, and this is 250 base pair, and now this is called fragment size. Okay, so we did all this process in order, if you try to think a little bit the whole process, we start with a hypothetical shearing size, and we did all this process to find from the data the fragment size. And since we have now the fragment size, we can proceed and analyze the regions and try to find the interesting peaks. So this is the first video, and now we'll go to the next one to continue the process.